Hi, my name is Alex. I'm a senior majoring in MCB and CS. I joined Dr. Bellin's lab in my freshman year after getting interested in microbiology through our first year seminar course. To me, bacteria seemed all the same, but after taking the FYS course, I realized that every species can differ in so many ways. So I decided to join a microbiology lab to learn more about bacteria. The Bellin lab studies Radiorhizobium diazooficans, a nitrogen-fixing bacteria that forms symbiotic relationship with legumes like soybeans. When I first joined, I quickly learned that non-model organisms come with challenges never mentioned in class. Beta efficients, for example, readily develop antibiotic resistance and discard exogenous DNA. So you can't test genes of interest on a plasmid, but you have to integrate them into the genome. But even that's a complication because methods like electroporation, which works really efficiently on E. coli, does not work on our species, so forcing us to actually use a more convoluted conjugation-based method where you put the plasmid first into a separate species of E. coli and get that E. coli to then give it to our wild-type species. Finally, there was no easy deletion methods, and we had to rely on primitive and very challenging recombination-based method. Interested in solving this problem, I started implementing CRISPR-Cas9 lambda-res systems in our species. I spent about a year designing and doing assembly and optimizing assembly, but ultimately discovered that the only available inducible promoter in our lab simply cannot be cloned, and it was also leaky, so we cannot use it for proper CRISPR-Cas9 system implementation. Currently, I am now working on identifying better inducible promoters so that if successful, we can finally have a more robust inducible promoter system, and also make Cas9 lambda-res systems more of a reality. After that setback, I took an alternative approach. Instead of optimizing genome editing, I tried to optimize plasma retention. To do this, I turned to histidine oxytrophy, deleting a HisF gene so that cells would require external histidine to survive. The deletion was successful, but when I tested plasmid carrying HisF, one promoter was lethal and the other caused plasmid to integrate into the genome, preventing me from testing plasmid retention successfully. Now, this does imply that this marker is very robust, as I was able to see that cells die without histidine, and with the endogenous promoter, all colonies actually carried our plasmid, but they just preferentially integrate into the genome. So now I'm redesigning the testing plasmid with a different promoter that is weaker than the constitutive promoter I use and does not share homology with the genome. Through these projects, I've gained independent research skills, and I really refined my wet lab techniques as I had to do processes again and again and again myself. I learned to maintain detailed notebooks and strategically plan around B-diazoofiction's slow growth rate. Our species actually takes 3 days to grow in liquid culture and 7 days to grow on plates, so I really had to plan around that to prepare for experiments, also prepare for unforeseen circumstances. Finally, I've really practiced designing and troubleshooting experiment and learned to use failure to optimize every single details of experimentation. My biggest realization is that unlike other areas of life, trying harder simply does not equate success in research. Effort doesn't guarantee results, as sometimes the cause of the failure is not you, but it's actually biology. And so even though that's research has been challenging, I've really found it exciting whenever I make a small incremental progress. And it's really exciting to just know more about biology and know that every single work is building towards something. And I'll definitely pursue this as a career in the future. Thank you.